Okay, good day, everyone, and welcome to our webcast, Transforming Nutrient Removal in Water Resource Recovery Facilities through Suboxic/Low DO Treatment. This webcast is part one of a three-part webcast series on this topic. Part one, we are calling Nutrient Removal with Minimal Aeration Works, exclamation point. Introduction to Low DO/Suboxic Process Operation and Microbial Fundamentals. Part two will focus on aeration requirements, and part three will focus on a full-scale demonstration at Los Angeles County Sanitation District's Pomona Water Reclamation Plant. So part two will be November 5th and part three, November 19th. But for today, we're going to focus on operation and microbial fundamentals. My name is Jeff Moeller. I'm a Director of Research Services at the Water Research Foundation. I'll be serving as the moderator for today's webcast. I'm going to go briefly over a few housekeeping items, as well as a high-level overview of the agenda before I hand it off to our first speaker for today. So just in terms of housekeeping, you can submit questions through the question box at any time. We'll do a Q&A at the end of the presentation, so just keep the questions coming throughout the webcast. Slides and a recording of the webcast will be available at www.wrrf.org. You can download the slides now and access a link to a project page under Event Resources at the bottom left of your screen, so those resources are available to you. A certificate of completion will automatically be generated after the webcast. If you have any questions, you can contact Michelle Suazo at the email shown on your screen there. And then we encourage you to please stay on until the end to fill out a quick survey with your feedback on the webcast. So in terms of our agenda today, after our welcome and housekeeping, our agenda is broken out primarily into two parts. The first part will be an introduction to low DO/Suboxic process operation, and that's going to be given by Tanya Rausch-Williams of Metro Water Recovery. And then the second part will be on microbial fundamentals, including nitrifier adaptation and biological phosphorus removal improvement. And that's going to be presented by Dan Noguera from the University of Wisconsin-Madison. And then, as I mentioned before, at the end, we'll set aside some time for any questions that you have, as well as a little bit of discussion. So we have two primary presenters today. Our first will be Tanya Rausch-Williams, who is the Chief Innovation Officer at Metro Water Recovery. And then she'll be followed by Daniel Noguera, who's a Wisconsin Distinguished Professor at the Department of Civil and Environmental Engineering at the University of Wisconsin in Madison. Their full bios are available on the platform. There's a tab with their bio information if you'd like to read that. And I think without further ado, I will turn it over to Tanya.
Thank you, Jeff. Can you hear me well?
Yes.
Wonderful. Good. Well, I have the pleasure to kick us off here with an introduction to this project and low DO or suboxic process operation. I want to say much of that work that we started with this DOE project about 2.5 years ago was done when I was still with Corollo Engineers. So just to kick us off, I'd like to summarize sort of the value or the value of low DO suboxic treatment in a nutshell here. We often talk about low DO treatment for energy savings. And I showed here a graphic that took a great time to compile during this project that we did over several years. We'll talk much more about the details of this graph and what we did at Pomona over the next webcasts. I just want to highlight that this graph really greatly shows energy savings that low DO and suboxic treatment can achieve by what you see, just about 30%. From where we started out, about 1,500 kilowatt hours per mg treated to less than 500. That was a great saving, even more than what I've shown here, 25 to 50% cost savings. But really, for many utilities, the energy savings are second to the nutrient quality that they need to achieve to discharge the effluents. And so what we are going to focus on, and we'll show you with the 3 webcasts, really all of them, is that not only energy savings are achieved, but that we greatly improve the effluent quality. And that is not just with denitrification, where we can get nitrate concentrations below 5 milligram per liter in just really BNR processes, but also very consistent nitrification. In fact, we're going to show you in the 3rd webcast that we have that we were able to improve nitrification operating at lower DO conditions. So that is really unexpected for many utilities that is possible, and that we are able to achieve in utilities that are not even designed for phosphorus removal, biological phosphorus removal, that we are able to achieve with low DO treatment, very consistent enhanced biological phosphorus removal, well below 1 milligram per liter. We also want to highlight in this series of webcasts that this is not a operation that requires very sophisticated operation staff or PhDs running facilities. In fact, low DO treatment has been implemented, especially at facilities that are either understaffed or don't have the resources to greatly, pay attention to the process. And there are reasons for that we'll discuss. So really this webinar hopefully finds a lot of open ears and interest from utilities across the country because it improves, it lessens O&M costs, it improves effluent quality, and that at a smaller or less capital infrastructure that we should capitalize on when we design new or modify existing BNR facilities. I want to step a few years back when we started this process and just how was the lay of the land in North America at the time, and why did we set the focus of this project the way we did. So going just back about 5 years ago, if you were working in low DO treatment already at that point in time, you know that we had several facilities in the US that operated since years in low DO operation. Typically, and most commonly, those were oxidation ditches. And we had them across the country in warm weather and in cold weather zones. Here, just an example from City of Montrose, a facility we worked with here in Colorado, a smaller facility that is facing nutrient requirements for nitrogen removal and for phosphorus removal. And really at the time, they had difficulties even nitrifying their facility. And so for an oxidation ditch facility, really with very low investment, about $200,000 that included some additional monitoring system for nutrients, nitrate, and ammonia, and then some fees for external services, consulting services, and largely with the help of the operators in very short amount of time, we were able to reduce the DO, the aeration, into the oxidation ditch. And with that, you see on the lower graph an improvement on nitrification and greatly of denitrification. So that was a great achievement from that utility, but it also wasn't that special. Many utilities across the country on oxidation ditches operate in this facet. What was unusual some years ago was to do the same kind of treatment and achieve that in plug flow aeration basins. And why has it been so difficult to operate low DO facilities in plug flow aeration basins? Really, the majority of our facilities are plug flow basins, operate plug flow BNR configurations. So one is that really the diurnal influent flows and loads are much more pronounced as we are operating in basins with shorter HRT. Oxidation ditches, you could operate at 24 hours retention time with very long SRTs. That's not a luxury we have at flow-through aeration basins. And so oftentimes utilities see uneven load distributions be among the aeration basins. So it's difficult to control the air. And oftentimes utilities do have effluent requirements specifically for ammonia. And we see breakthrough of ammonia in the effluent, and that makes operators nervous when these breakthrough and spikes get higher as we lower the DO. So, and all of that, in fact, we observed at the field site, the full-scale field site at Pomona, that we'll focus on in the third webcast. And just exemplarily, I show you here a graph to the right, where you see that this facility has very short HRTs, short SRTs. We see diurnal breakthrough of ammonia with spikes up to 10 milligram per liter. And so that these operators were just as nervous about embarking on a low DO adventure with us 2.5 years ago. So we'll show you that as we move through specifically the third webcast. What a development that also happened several years ago in the US is that we started, and several vendors started, to look at enhanced aeration control systems. And that's the emphasis really in the second webcast, and was the second piece that we pulled in when we proposed this project for DOE. We have aeration control systems now available that allow us to much tighter control DOs in various control configurations, whether ABAC or DO control, AVN, but much tighter than it was possible in the past. So what you see here on the left side, again, an example just from this study, but many facilities operate this way, where you see large fluctuations of the actual DO around the set point. This facility operated at Pomona with various DO set points throughout the day just to better buffer off the load. And with the implementation of the enhanced controller that you will hear a lot about in the second webcast, you see the benefit that can be achieved, very small amplitude changes around the set points. We essentially just mirrored exactly the same control logic, the same control set points that the facility operated earlier, but with a different controller. And so really these two aspects, that we have control systems that allow us to really control the DO with the knowledge about how we need to operate and adapt the microbiology towards low DO. That's what we proposed to DOE to embark on this project and try this out at a facility that was, flow through, a low, short HRT, requirements for effluent nutrient removal, etc. So here, just very briefly, the DOE partnership with all the different partners that helped us along this project. I'm not going to go through in detail, but acknowledgements to DOE to fund this work. One of the first efforts we did while we prepared for pilot testing, full-scale testing, was we did a facility survey of low DO facilities. And many collaborators helped in North America to identify and help share data from those utilities with our team. Here's a summary of about 9 facilities of the ones we looked at that we summarized. You see, we had different utilities with different types of BNR processes. We started categorizing the type of low DO treatment or configurations that I will introduce in the next slide. You see that here, whether we call them fully or partial suboxic. Some of these were oxidation ditches. We call them here complete steered tank reactors, CSTRs, and several were flow-through reactors. And then we asked them for long-term data, whatever they were able to share with us while they were in this low DO operation, to draw some just empirical assessments on process performance and effluent quality. So I want to show you a couple of slides that just group how different configurations that utilities can employ to transition or work into low DO operation. On the left side, you see conventional aeration. So the light blue zone will always indicate an operation at higher DO concentrations, let's call it higher than 1 or 1.5 milligrams per liter. And on the right side, I show a tapered aeration. Many of you will be familiar with that. So this is when utilities take either the very front of the basin where they're operating at depressed DO conditions, or at the very end of the basins where the DO is tapered off maybe not to recycle and result in oxygen poisoning. So that is one step for low DO operation. Another one is when utilities implement ammonia-based aeration control. That control will depress the DO as much as possible unless the ammonia set point is exceeded, at which point the DO would be increased, maybe to concentrations above 1.5 milligram per liter. But oftentimes, these utilities operate either spatially or temporarily at different parts of the aeration basins in low DO conditions. And let's define low DO here operational as average concentrations of less than 1 milligram per liter. Some utilities wanted to ease into low DO and maybe just try to operate one of their basins in a low configuration while operating the complete BNR process as a single sludge system. So you may find at these utilities that one basin is operated at a depressed DO of less than 1 milligram per liter, while the other basins are higher. So the biology would be exposed to low DO and high DO conditions. And then I talked about already the oxidation ditches, where we have across the US examples that are operating at very low concentrations of DO. You see here a facility that never operates higher than 0.5 milligrams per liter, achieving full nitrification and very good BNR treatment. And very few facilities that we were able to identify, I think we only found 2 at the time. We did that about 2-3 years ago. 2 facilities we were able to identify that operated in a plug flow operation with ABAC control. You see here one example where the concentrations were consistently in all aeration basins below 1 milligram per liter on average. So very few facilities like that. I want to show you in the next 3 slides, or maybe 4 slides, just empirically data from these facilities. I said we asked them for long-term data and plotted this in terms of these box and whisker plots, just to get an empirical idea where do they operate in terms of nitrification, nitrogen removal, phosphorus, and sludge settlability. And so what you see here is on the left side will always be DO. We put a target in of 0.7. We define that just operationally as if they are largely with a data set below 0.7, we call them a low DO facility. And then on the right side in green, the data is effluent ammonia concentrations. And we put in a target here of less than 1 milligram per liter. And what you see is that a number of facilities that are fully suboxic, either in oxidation ditches, for example, look at facilities C and D, or partially suboxic in plug flow, are able to achieve very good nitrification at low DO concentrations. And interestingly, also when you look at the high DO facilities, look, for example, at facility I, and we know that, we have facilities that operate at high DOs and still have difficulties with nitrification. And so we'll talk about that more when we talk about the microbiology and the experience we have at Pomona and other facilities, that there is actually a biological mechanism that makes it simpler and more robust to nitrify at low DO condition, even though that sounds first counterintuitive from traditional activated sludge, treatment as we learned it traditionally. Here you see the same type of data represented, but now with inorganic nitrogen. And again, you see facilities C to F that are operating fully suboxic or partially suboxic in plug flow, achieve very good denitrification, oftentimes below 5 milligrams per liter of ammonia and nitrate combined in the final effluent. And that is traditional because of we are allowing for simultaneous night-to-night to happen. Interesting is also, and Dan will talk about that here in the second half, that we see facilities with very good phosphorus removal, and we have examples of MLE facilities that don't even have an anaerobic zone that are able to get very efficient biological phosphorus removal with phosphorus concentrations of less than 1 in the effluent. SVI performance is a mixed bag. We know of facilities that are operating under low DO or suboxic conditions and that do not have elevated SVIs, and we know facilities that do see settleability issues. They may have large clarifiers or enough capacity to cope with that, but that is something that we have looked closer into in this project as well, and we'll discuss. So really, why talking about low DO and why introduce the term suboxic? I want to show just exemplarily, and we'll get more into that in the next webcast. But here's some data from Pomona, which shows how we over time reduce the DO conditions in their two aerated zones in their basins, grid B and C. And so you see that over time we reduce the DO from up to 3 as a peak down to 1 and about 1 and 1.2 or 1.3. And it's interesting for us, as we did this project and really needed to figure out operationally, how do we create guidelines for the industry to transition and adapt into low DO conditions? Does this adaptation actually happen continuously, or are there certain phases or boundary conditions in the DO ranges where we see the microbes adapt faster? And I overlaid here for the same time period the effluent ammonia concentrations, because there is a variability diurnally. You see that this data looks kind of smeared, but that's just the diurnal variability of ammonia in the effluent. And specifically, I don't have a pointer, but look at August of this year. So towards the end, we suddenly saw a decrease in ammonia that we pay attention to in the third webcast specifically. But once we had the biology exposed for a certain amount of time or for an extended amount of time to very low DO conditions, at that point we were with a 50th percentile at about 0.7 milligrams per liter. And we just slightly increased the DO. Ammonia essentially disappeared. And the peaks that the plant has always observed in the past suddenly vanished. So this indicates that there are boundaries or redox conditions in low DO operation that actually trigger the biology to a shift in performance. We'll talk more about that later. And so that's why we've introduced in this project the term suboxic. This is really not a new term. It has been used in soil sciences for exactly that purpose, to describe biology, microbiology that's adapted to low DO concentrations. And you're all familiar with aerobic, anoxic, anaerobic conditions. And so we're postulating that there is actually a new or an additional redox condition that happens in the low DO range. Here, operationally, we define it at 0.2 to 0.7 milligram per liter. And that refers to the average DO in the aeration basins over the course of the day, that if you're staying below that, biology seems to adapt and find a new equilibrium for nitrification. So just 2 more slides before I turn it over to Dan as a second speaker, just to give you an outlook of what to expect in the next webcast and what we focused on in this project. Really what was important for us is not just to have one more field site to show this is doable, but also to tease out what operational and design guidances can we deduct from, yes, our field site at Pomona, but also the surveys we did at the other field site. What are the commonalities that help other facilities transition faster and in a controlled fashion into low DO? And how can we design facilities, new facilities, or modify existing facilities to equip them with the infrastructure to operate safely at low DO conditions? I'm not going to go over all of these points, but many of those will be hit on in the next webcast. So really, to give you just an outlook, this first webcast, Dan will focus on the microbial fundamentals. What do we understand of low DO and suboxic organisms to do, and how do they adapt? And in the second webcast, we talk about the control and the aeration system side. So that's the infrastructure side. How do we make that right-sized, fit, and the controls tight enough so that operators have the tools and the flexibility to really be in control of the process as they're driving it down to low DO concentrations. And then in webcast 3, we bring it together and talk about the Pomona field side and how we all implemented it there and went in very systematic steps down to low DO and the lessons learned for other facilities. So with that, I'd like to turn it over to Dan to give us the introduction into the microbial fundamentals.
Thank you, Tanya. So I'm going to talk about what we know about the microorganisms in here, and I'm going to give you a view of 20-some years, 25 years doing research on this area. And here you have a timeline of some of the work that we have done, some of the industries we have worked with. So I don't know if my video is showing or not, my camera is showing or not. Michelle can look into that. But we have been working on this low DO area for a long time, lab scale, pilot scale, food scale. And I mostly focus on the genetic approaches to identify microorganisms. And with this in mind, there's a couple of discoveries that have occurred throughout the years that we have been doing this. In 2005, ammonia oxidizing archaea came into play. We didn't know that when we got started. In 2015, Anamox or common moss was discovered. Anamox was known from way before that. So it's every 10 years. I don't know what's going to come up in next year in terms of something, but I don't think we know everything we need to know about who is doing nitrification in these processes. So in terms of the molecular tools, when we started, the typical tools that existed was doing FISH, or in situ hybridization, or clone libraries. Later on, we were able to do PCR, quantitative PCR, to look at functional genes within the organisms. And nowadays, we are on the stages of doing whole genome sequencing with a couple of platforms, Illumina, PacBio. So what I'm going to talk to you today is mostly about the first two. We're still working on the last part of it, but I'll give you an overview, more of historical overview of this. To begin with, what is nitrification, or what are the microorganisms that are doing nitrification? So you have the typical ammonia oxidizing bacteria, and now ammonia oxidizing archaea doing ammonia oxidation to nitrite. Then nitrite oxidizing bacteria doing nitrite oxidation to nitrate, and you have the typical denitrification done by several heterotrophic organisms that bring back and produce nitrogen gas from that. Anamox, it's another organism that is there, probably not a whole lot in activated sludge, with ammonia and nitrite getting converted to nitrogen gas in an anaerobic environment. Then you have this new discovery of COMAMOX, complete ammonia oxidation, which are nitrite oxidizing bacteria that acquire the ability to oxidize ammonia to nitrite. So in many of these original ways of detecting organisms, they were identified as nitrite oxidizing bacteria, and that will come up later in the presentation. Like I said, I don't think we know everything. So there are probably pathways that we haven't identified on how nitrification is occurring, possibly in these very low oxygen environments. Phosphorus removal, enhanced biological phosphorus removal. We know that when we cycle activated sludge between anaerobic and aerobic conditions, in this case, low oxygen conditions, there are these organisms that we generically call PAOs, polyphosphate accumulating organisms, or DPAOs, denitrifying polyphosphate accumulating organisms who are playing a role in these treatment plants. So anaerobically, they both take acetate as their main organic substrate. They release phosphate. That's why we see spikes of phosphate coming out in the anaerobic stage. And then when there is oxygen available, oxygen acts as the electron acceptor for the growth of PAOs. The nitrite, the oxidized nitrite species as the electron acceptors for denitrifying PAOs with the same outcome of phosphorus being consumed, microbial growth occurring, and then you get your treatment occurring for phosphorus removal. So microbiologically, we have known for a long time that we can really establish very suboxic nutrient removal without any problem. This is an experiment in the lab with a sequencing batch reactor, the typical observations where, as shown there, where we feed the organisms acetate in the anaerobic stage, all that acetate gets internalized into the cells, phosphate gets produced, and then when you turn into oxygen, in this case was very low oxygen, our threshold was 0.2, but during the active consumption of the organic matter by the microorganisms, so the first 4 hours here, or from 1.5 to 4 hours, the oxygen was pretty much undetectable. It was 0.05 or something like that. And then you see complete removal of the phosphorus. You see nitrification. You see no accumulation of nitrite or nitrate. And then when all the organic is consumed, then this threshold of 0.2 milligrams per liter of oxygen starts taking place. We had an on-off switch, and then so the oxygen fluctuates around 0.2. When all the ammonia is exhausted and there is no more oxygen demand, that's when we started seeing more oxygen. So this is an example of complete nitrification, complete denitrification, complete phosphorus removal, maintaining oxygen at less than 0.2 in general on the overall thing. So we have known that in the lab for a long time. One of the first experiments that we did was 25 years ago, looking at nitrification in one of these oxidation ditches. This is the Marshall Water Resource Recovery Facility in Wisconsin, where they had a very low oxygen in the outer channel, close to undetectable levels, and so on. And they had 50% nitrification in that channel. So we wanted to compare the microorganisms doing nitrification in that plant with the microorganisms doing nitrification in our conventional UCT type process in Madison, Wisconsin, where you had, at that time, you had very high aerobic dissolved oxygen concentrations in the aerobic stage. So we took samples from both and we did cloning, just looking at the 16S RNA gene of these ammonia oxidizing bacteria, and then you get the tree of all the different organisms that might be identified in there. So I'm going to color code the ones that we found from the treatment plant operating at high oxygen, the ones labeled as 9 Springs there, where most of the cluster was within the traditional Nitrosomonas europaea organism. And the ones in Marshall, which had this 50% nitrification occurring at very low oxygen, most of the clones were on the Nitrosomonas oligotropha side of this phylogenetic tree. So that was our first observation that there is a change on the Nitrosomonas organisms between low oxygen and high oxygen. Another experiment that we did years later was taking samples from this high oxygen treatment plant, local treatment plant here. And at that time, we were working with American Water and a treatment plant that they had, a membrane bioreactor in Jefferson's Peak wastewater treatment plant in New Jersey. So very high concentration of solids, but they were operating already with very low DO for several years when we started this interaction. And then what we saw, this was Colin Fitzgerald's master's thesis. We decided to take samples, put them in these reactors, lab scale reactors at very low oxygen, 0.2 milligrams per liter in each case, and observe over time what would happen. Would they start nitrifying? Would they not nitrify? And so on. So here is the graph from samples taken from the high DO situation where ammonia, the blue here, is entering at a concentration that is about the same concentration in our wastewater treatment plant. It took us about a month and a half before we started seeing real nitrification happening. And then when you saw nitrification happening, you saw accumulation of nitrite. And then at some point, the nitrite was also removed, and then you got nitrate production. So in this situation, we were adapting a community that was operating at high oxygen to low oxygen, and it took this long over time. Compare that to the other treatment plant, the Jefferson Peak treatment plant, where immediately after, when we started the process, nitrification was occurring. There was a nitrate already being produced. There was ammonia or nitrite, very little concentration of nitrite, and then it was consumed. So both stages, ammonia to nitrite, nitrite to nitrate, were effectively happening at low dissolved oxygen. So then we asked, can we figure out what's doing this? What are the ammonia oxidizing organisms involved in this? And this time we were doing quantitative PCR, looking at the function, looking at the ammonia monoxygenase gene. And this is a summary of what we found. On the sludge from 9 Springs, we saw the exact same thing of an enrichment of nitrosomonas to the nitrosomonas oligotropha group of organisms. That was what we had seen before, and that was consistent with that. The sludge from the other plant, the one that had been operated for many years as a low DO condition, there were no ammonia oxidizing bacteria that we could detect, nothing. We also looked for ammonia oxidizing archaea, another organism that can do ammonia oxidation, not present on either plant. So that would not explain ammonia oxidation on either one. And then common moths. This was at about the same time that common moth was starting to be described, but when we did the experiments, we didn't know this fact that common moths existed. What we saw was a difference on the accumulation of nitrite oxidizing bacteria. And in the Jefferson's Peak, the low DO sludge, Nitrospira moscoviensis related NOBs were the ones that were prevalent. Now, Nitrospira moscoviensis related NOBs are the ones that are closely related with what we know to be common moths organisms right now. 16S is not sufficient to differentiate that. That's what we were doing with nitrite oxidizers. So unfortunately, we didn't go as much as being able to identify exactly whether there was a common moths organism or not. But we know that in the other process, the one that came from the high DO environment, we enriched for a different group of Nitrospira organisms, the ones associated with Nitrospira defluvii, which is not the group of NOBs that contain the common moths organisms. So there is a strong evidence here that common moths could have been the organism that was doing low DO activities on this Jefferson's Peak plant. And also that we didn't enrich for that type of organism. We didn't enrich for common moth type organisms at this lodge here in Madison when we subjected to low dissolved oxygen. We have done also pilot scale experiments. This is the work of Natalie Beach, in which we operated pilot plants now subject to the variations of temperature that we will experience here in Madison and so on. And this experiment was lowering the oxygen slowly, as similar type of experiments that Tanya showed earlier. And what you see here is in terms of the 16S signatures, a change on the group of ammonia oxidizing bacteria that were enriched on the plant. And the pilot plant that was subjected to low DO compared to the high DO plant, the full scale plant, there is an accumulation of a different group of ammonia oxidizing bacteria. Again, those are Nitrosomonas oligotropha. One of the things that you see on the right-hand side here is an analysis of the colonies that are formed. Ammonia oxidizing bacteria are known to grow in this microcolonies inside of the flux, spheres of a certain diameter. And the question we asked was, when we subject this to low DO operation and the plant is efficiently working at low DO, how does the size of those colonies look like compared to the size of these colonies of ammonia oxidizing bacteria in the full scale? And that's the plot that you see there. In blue is the full scale high DO, in red is the low pilot plant low DO, and there is a significantly different size of these colonies, whether that's because it changed to Europea from Nitrosomonas, it changed to Oligotropha from Europea. That could be an intrinsic change on the conditions of these organisms, or the low oxygen environment creates a difficulty in the diffusion of oxygen. What we know is that translates to a reduction on the kinetic parameters for low DO nitrification. The KDO in our case, 0.3 compared to the full scale plant of 1.4. And through this project that these webinars are describing, we saw the same things happening. So it's a real change that happens on the kinetics with respect to oxygen. So that's what I'm going to tell you about nitrification. I'm going to spend a few minutes telling you about biological phosphorus removal. And the story is not that interesting from the point of view of nothing really happens. On treatment plants in which we have really good EBPR operation, lowering the oxygen does not negatively affect them. This is a case, this is a diagram of the full-scale treatment plant here in Madison, where we did a full-scale experiment of going from high DO to low DO, and we did normal operation. You can see there the oxygen concentrations at different parts within this plug flow reactor. And then when we did aerated anoxic operation, or lowered this, or suboxic, we call it aerated anoxic at that time, oxygen's below 1. And then we did a step feed operation too in there. What happened with the phosphorus when we did this? That's the graph of phosphorus during normal operation, during this low oxygen operation, during a step feed operation. Not negatively affected. We had excellent EVPR. We continue having excellent EVPR when we lower the oxygen. If you plot the data in a different way in terms of the amount of the phosphorus removed per unit mass of VSS, what we see actually is an improvement in the rate of phosphorus removal when we subjected this to low oxygen conditions. So whatever organisms are in there doing EVPR, they are not negatively affected by lowering the oxygen, which is great, because then you can focus on the problems that might be generated on the slow transfer from for nitrification, and kind of forget about phosphorus removal. It's going to work for you. So I'm going to conclude. The work that we have done, large scale, pilot scale, full scale experiments, demonstrate that suboxic nitrification is possible. Looking at that from an academic perspective, I can set up laboratory experiments. It works great. We add a little bit of practicality with the pilot scales, with the variations on temperature, the variations on flows and all that stuff. And we also see that it kind of works well. And then this project is moving on to the full scale. On which organisms do low DO nitrification, I don't have a strict answer there. Nitrosomonas oligotrophan, we know it does. And in treatment plants in which common MOX doesn't become prevalent, Nitrosomonas oligotropha seems to be fine doing this low DO nitrification. There are other treatment plants in which it appears that common MOX are the organisms that are doing low DO nitrification. And biological phosphorus removal is compatible with the suboxic operation. There's no limitations to the process. I know the phylogeny, or the amount, the different types of organisms that are being claimed to be related to enhanced biological phosphorus removal is increasing. in recent years. In our experiments, we have always seen Candidatus accumulobacter phosphatis as the main organism that does ABPR. And I could tell you more about the different types of accumulobacter phosphatis and what we have seen with low and high oxygen, but we are running out of time, so I am going to stop here. Acknowledge that a lot of students have been involved in these activities. Madison Metropolitan Sewage District has been our main partner on this, and many different industry collaborators. So thank you very much for your attention. We'll be happy to answer questions.
Thank you, Dan. We really appreciate you walking us through the microbial fundamentals of this process. And I think coupled with Tanya's earlier introduction to low DO suboxic process operation gives us a nice introduction to this topic. So now I'd like to open the floor to some questions. We have had some questions come in while our presenters were talking, and I encourage folks to continue to add questions to the queue, but we'll start to tap into some of those. So we did have a question come in asking, so far, what makes a plant a good candidate for low DO application? For example, what temperatures or process types or any other types of things that might make it a good candidate?
So from our perspective, we have seen temperature is something that you have to be careful with. If the temperatures go too low, in Madison, the first experiments that we did, nitrification was effective. There's not enough safety factor when we do low oxygen and low temperature at the same time. But you can find conditions in which you can operate at effectively. I don't know if Tanya has other thoughts.
Yeah, I mean, I think there is less, I hope from the examples that I showed that there is less maybe focus on the configurations, because we find low DO operation in various BNR configurations. I do think there is still a question outstanding on temperature, and several of the questions here focus on temperature. So I want to quickly summarize what I think we know, and you'll see that also in some of the next webcasts. So we have facilities here in Colorado, and I showed that quickly on the map when we did the survey. We have facilities here specifically that are operating in the Rocky Mountains. They have temperatures, wastewater temperatures of 10 degrees Celsius. So that's almost as low as it gets. And they operate at, I actually showed the data from one of the facilities with DOs of less than 0.5 at any point in time, not average, any point in time. And they nitrify completely, denitrify perfectly, and remove phosphorus. That is an oxidation facility which has a long SRT. So the question of SRT remains: what adjustments are needed at low temperatures? At Pomona, we'll talk about that more in this third webcast. But at Pomona, we were very intentional to not change the SRT and we didn't change the HRT. We didn't lower the load coming into the facility. And if you remember the graph I showed towards the end, when we started at that facility, you could call this facility on a nitrification scale to be overloaded, because the daily spikes of ammonia coming out of the facility hit 10 milligrams per liter frequently. And it just allowed the facility to essentially sail under the monthly average, the monthly max that they needed. So at high DO condition, that facility was essentially at the capacity limitation. We didn't change any of that, and we are now operating well below that. You saw the improvements. So I would say and the HRT of that facility is quite low. It operates on the aerobic side between 3.5 and 5.5, depending on the diurnal peaks coming in, hydraulic peaks coming in. So that's on the lower end of HRT for aerobic conditions. And the SRT at that facility, we kept steady at 6 days. So that's not as low as we probably could get, but yeah, that's a long-winded answer. I covered a couple of the questions, Chip.
Yeah, no, that's good. Thank you. Yeah, we had one question come in asking about what is the risk of exceeding permit levels while transitioning, or, maybe it could be rephrased, or is there ways to kind of minimize, the risk of exceeding permit levels, and how to go about that?
Dan, I could answer. You could go first. So, I think so it depends on the permit limits. Okay, we have facilities that have daily max ammonia limits. I know facilities that have daily max ammonia limits of less than 2 milligrams per liter. So you got to pay every day attention exactly what your ammonia does any time of day to not exceed that. We have facilities that have max month limits. In the lower in the lower single digits, but that's achievable. We really emphasize in the sequence the relevance of good DO control, because with the DO control systems we have, the advanced control DO systems we have today, it is possible to transfer facilities from high DO to low DO in a safe manner and maintain the same nitrification performance or get better. As Dan said, I agree with him that we were not worried about denite or phosphorus removal, because that seems to just follow suit and get better. We really focus on nitrification. And so I do emphasize how important the DO control is. I showed you that photo, the graph at the very beginning of the facility when we started operating there. Take a facility that has very high amplitudes of DO, switching between 0.5 and 2.5 or 3 milligrams per liter every day. If you don't fix that control issue before you even start, and you lower the DO set points down by 0.5 milligram per liter, you're having low DO condition or no DO conditions and very high DO conditions within hours at a utility. And that creates filament issues. It does not put the consistent pressure on adaptation on the biomass, and we saw at phases in Pomona that it can result at high at ammonia breakthrough. So that needs to be controlled. But I would say there is a way to set this up so that you're not running into permit risk.
Thank you. We've had a couple of questions come in related to HRT, and including one, what is the range of HRT to be considered challenging for low DO operation?
Yeah, Dan, please jump in. My experience is empirical from utilities that are operating like that. As I said, at Pomona, the aerobic HRT at the time of breakthrough of the high ammonia concentrations at the diurnal in the effluent is at around 3.5 days. So we were for a while wondering, is it the HRT? Why we're not getting lower as we lowered the DO? Why are we not able to reduce? We stayed pretty constant with the spikes or the maximum ammonia concentration over the day. We did not change the load. We kept it. We stayed with it and lowered very slowly the DO concentrations. And then, as I showed you, we had that breakthrough essentially where we were at low HRT, still able to essentially knock out the diurnal spikes. So there was definitely an acclimatization of the biomass that at lower concentrations was able to fully nitrify the spikes coming in. So what we have not done, and that's difficult because we're at the end of the project when we have the process right now in a really cool, spot, it would be really helpful to increase the flows, lower the HRT farther and see where is actually the boundary condition, lower the SRT. Right now we're going into winter, so we're going to keep it the same. But what is the play or the sensitivity with SRT at the full scale condition where you start seeing washout? To my knowledge, and please, if anybody, we have a lot of people, and I'm sure some of you have experience on that. If you want to post your experience with that and know what boundary conditions you have tested and where you saw limitations, I think that would be great. I'm not aware that we have done that systematic testing yet at full scale.
Thank you.
Yeah, same for me. We have done HRT. All the reactors have been set up to what the local treatment plant uses for HRT. So we don't have really experience varying the HRT.
And this is a question for both Tanya and Dan. Any thoughts on impacts of low DO on settleability, SVI, and what, if anything, can be done in the field to address that concern?
Dan, do you want to go first?
So that's probably the main issue from our perspective, that the SVIs go up a little bit under certain conditions. One of the things we tried to do to take care of the low rates at low oxygen and low temperature was to increase the SRT. That wasn't that good in retrospect because we got to solids concentrations that the sludge wouldn't settle. So the SVI seems to be a bigger thing. I think the industry has now different ways of dealing with poor settleability sludge and additional things that could be tested. But that's more of a physical phenomena, in my opinion, that will need to be tested in a different way or answered in a different way.
Yeah, as you said, and there are selectors, right, that we can use, whether they're internal selectors or external selectors, to select out filaments. I would just say, I think that verdict of low DO and high SVI is still outstanding. I think as we are paying much more attention to aeration control, and we know what low DO filaments, what causes them, and one factor that causes them is fluctuations in DO, very rapidly fluctuations in DO, sometimes low, sometimes high. That's just one factor. So we can minimize that. What we saw at Pomona, we looked at that very carefully and did sludge analysis and SVI analysis. So what we saw there is that as we transitioned from high DO to low DO, we did see higher SVIs, specifically around that. It seems to be a boundary condition to get from somewhere 1.3 to below 1 milligram per liter. So we saw sustained high SVIs, nothing unusual. The plant has seen about 200 during winter conditions. So that was not unusual or not unmanageable, but it was seasonally obvious that it was different. As we dropped to low DO conditions, so around 0.7 milligrams per liter, the SVI improved, and we were the same or better compared to previous same season SVIs. So there may be several things at play that we need to document at facilities and find best management practices to manage that, but also really look at low DO condition, not the transition phases, and what facilities are operating there at steady state conditions.
All right. Thank you. And I think we've just about out of time here. So I do want to thank all of our participants. We've got a ton of questions that have come in. We'll maybe do our best to follow up with participants on providing responses to those questions. There's more than we can handle in our time here on the webcast today. But we're at time, so this concludes our webcast. Thank you all for participating, and special thanks to our speakers, Tanya and Dan. We hope everyone will be able to join us for parts 2 and parts 3 of this webcast series, which will focus on the requirements around aeration. That'll be part 2 on November 5th, as well as the full-scale demonstration at LACSD's Pomona Water Reclamation Plant on November 19th. And registration is currently open for the one on November 5th, so feel free to sign up on the WRRF website. But we really appreciate everybody participating, and thank you all, and have a great day.